All right. Good day, I suppose I should say, to all of you from all around the world. Welcome to the RNA Collaborative seminar series for today. My name is Frank Slack, and I'm the director of the Harvard Medical School Initiative for RNA Medicine. And it's my pleasure to welcome you to this seminar series today. Today, we're going to have two seminars from our faculty. The first is going to be from Dr. Tim Yu. Dr. Yu received his degree from his undergraduate degree from Harvard College, and then his MD PhD from the University of California at San Francisco, and then did his internship, residency, and fellowship at Mass General Hospital and Children's Hospital in neurology and neurogenetics. He then became an assistant professor in pediatrics at Harvard Medical School and an assistant in the Division of Genetics and Genomics at Boston Children's Hospital. He's currently an associate professor. So just a reminder, if everybody could save their questions till the end, and then we'll give you a chance to ask your question. Either you can write them in the chat and I'll ask them, or you can unmute yourself and ask your questions. So without further ado, welcome to Tim.
Thank you so much, Frank. Thanks for inviting me to speak in this series. And it's a pleasure to meet many of you for the first time and say hello to several of those of you whom I've had the pleasure of interacting with before. The title of my talk is Prospects for Hyper-Individualized RNA Medicine. It's a little bit of a grandiose title, but I will perhaps humbly submit that this is work that really came across us unexpectedly through a story which I'll share with you. The outlines of the story may have been familiar to you as it's been covered rather widely in the press, but I hope that it will be instructive and stimulating to get some of your feedback on it. But since I am new to many of you, a little bit of background about my background. My laboratory at Boston Children's Hospital is in the Division of Genetics and Genomics. We work in the field of neurogenetics, and my training is in developmental biology, genomics, and human genetics. And the areas that we had really focused on previously were human brain development, genome analysis and interpretation, autism genetics, and with an interest towards genomic medicine. It was really only in the last 3.5 years that this interest in genomic medicine has really matured and turned into a series of projects that I'll describe to you today. But that genesis really all primarily began with a chance encounter with one very important patient to us, whom I'll share with you right now. So in January of 2017, we came across the story of a young patient who lived in Colorado. Her name is shared with you with the family's permission. Her name is Mila. And we learned through, of all things, a Facebook post that this young girl's family was reaching out for scientific help. Their daughter was 6 years old at the time and had suffered from vision loss, seizures, and neurologic regression after having actually a pretty exuberant and neurologically normal childhood before that. However, the previous 2+ years had been rife with these steady, insidious onset of these symptoms, and their family was looking for assistance with the medical genetic workup. Her physicians at Colorado had already made it a clinical diagnosis, a presumptive diagnosis. A skin biopsy performed by them by Dr. Austin Larson in their genetics division had shown that she had Batten disease. What it actually showed was a picture very much like this picture on the left. Electron microscopy of skin cells showed that there were fingerprint-like smudges present in her intracellular spaces. These represented lysosomal buildup of steroid lipofuscin, a semi-crystalline structure of mixture of protein and lipids that accumulates in the abnormal lysosomes in this condition. And it turns out that this was a form of one of 14 different subtypes of Batten disease. all of which involve recessive mutations in genes that are critical for lysosomal biology. And in the absence of their function, any one of those 14 will lead to lysosomal dysfunction and slow but insidious and progressive neuronal cell loss, especially in the central nervous system, the brain, the spine, and the eye. The symptoms of Batten disease fit exactly the clinical picture of this patient, and her particular subtype, CLN7, was extremely rare. Only 70 patients described in the literature. And CLN7 is uniformly progressive, always kills by age 11 or age 12. It's certainly of the many tragic pediatric neurology diseases, this is up there among the worst. And this patient, as I mentioned, her story may have become known to you because our experience with this patient was published in the New England Journal about 10 months ago. And it was remarkable to us and to others that we found ourselves in a position to go from first hearing about this patient in January of 2017 to identifying the underlying mutation, designing a drug to combat that mutation, prove that it was working in our laboratory, manufacture it, and then begin treating her with it within about a year. And just to hit the highlights of how this was done, because as I said, it is published work, so I can refer you to the article from last year, but I just want to hit the highlights because I'll use that as the basis for talking about prospects for scaling this type of work. Our opening in this case really began with our discovery of an unusual pathogenic mutation as the underlying cause of her condition. She had one mutation which was clinically identified, which was a vanilla mutation, a point mutation shown in that caused the missense mutation, and it was detected on clinical panel sequencing. However, her second mutation required whole genome sequencing by our group to uncover, and it turned out to be a DNA insertion of a SVA class retrotransposon. This is a mobile DNA element that's present in about 3,000 copies in the human genome. And this one had mobilized and landed deep in intron 6 of her gene. And it created, or actually activated, a pseudoexon, which is apparently latent in her genome and the genomes of probably all of us. But this pseudoexon is normally not used. We call it I6, but strong splice enhancers that were present at the ends of this SVA were sufficient to activate the pseudoexon, recruit its use, such that all splicing from exon 6 now ended, instead of productively in exon 7, landed non-productively into this I6 pseudoexon. And this hypothesis, which we had predicted from our genomic analysis, was proven experimentally in lymphoblast and fibroblast lines from the patient and her family. And it resulted in early incorporation of a nonsense mutation, which truncated the gene. The concept of developing a drug for her rested on our understanding of the mechanism of action of the RNA medicine nusinersen, or Spinraza. And many of you know this story super well. Back around Christmas of 2016, a few months before we met this patient, we were all reading reports of how this Spinraza was looking extremely good in phase 2 trials. And now, as you all know, it's FDA approved and has now been dosed to some 11,000 patients with spinal muscular atrophy. And this crowd needs a little introduction to the idea that this was a synthetic RNA that's given intrathecally 3 times per year, and it binds to and restores normal splicing of the SMN2 gene product. Our quest essentially was to make Spinraza for Mila, and just to cover again the highlights of how we did that, we first looked carefully at the region around our mobile DNA element insertion, characterized cis-regulatory sequences predicted from bioinformatic algorithms, and then to be splice enhancers in this region. And then we tiled those with a series of ASOs that we designed, manufactured, and then tested in skin cells from our patient. We were able to find several candidates in our initial designs that looked promising in terms of their ability to improve the ratio of normal to mutant splicing significantly by 6-fold in this diagram here. And we were able to do transcriptome profiling that showed that these application of these ASOs boosted levels of normal splicing in her cells from a baseline of between 7 and 11% up to 32%. And with this type of splicing molecular biology data in hand, we engaged a really dear collaborator, Joe Mazzulli at Northwestern, who was able to demonstrate functional rescue in patients' skin cells using our antisense oligonucleotide, showing that treatment of our patient cells, which at baseline contain these abnormally large white bubbles seen in this electron microscopy image here, representing lysosomes that were full of abnormal storage material. And we were able to shrink both the quantity and the size of these lysosomal storage vacuoles in our patient cells by administering our antisense oligonucleotide for only 24 to 48 hours, as shown in the green boxes here. And by a variety of other biochemical measures and enzymatic measures that Joe's lab had developed, we showed that it also, that this correlated with other measures of lysosomal function such as those shown here: autophagy, lysosomal enzyme trafficking, autofluorescence, and lysosomal mass. This type of work thrust us into uncharted regulatory territory because beyond the proof of scientific concept here, really the trick in this case was actually taking this and getting FDA regulatory and clinical buy-in to treat our patient. And we had to navigate a number of unusual new areas, namely the fact that there was no guidance previously written for cases where you were trying to develop a drug or bring into play a drug for a single patient. And this was truly a drug for just a single patient because our patient's mutation has not yet been found in any other patients with Batten disease before. And so just by illustration, finding a manufacturing partner who was able to make this drug quickly enough and to the appropriate safety standards required us to make up those standards. There had not been pre-negotiated criteria for releasing a drug under this type of circumstance. And even other simple logistical matters like the fact that we needed a very small batch of this antisense oligonucleotide. We needed only 10 grams of material by our back of the envelope calculations. Created logistical problems because many of the manufacturers who were capable of making GMP grade drug started at quantities significantly higher than that. Just as perhaps a fun example of the discomfort in which all parties in this space felt, this is just a list of the proposed and discarded labels for our drug these were all the proposals that went back and forth in a volley of negotiations between our hospital and the manufacturer. And I show this only just to illustrate how profoundly uncertain we and our partners were about exactly what the appropriate terms for what we were doing really was. Was this research? Was this for clinical use? Was this investigational? And so we ended up with a very carefully crafted label that I think just illustrates that this was new territory. But we did get through manufacturing and animal toxicology, and it took about only 3 months because we were under extreme clinical urgency. This is a disease which can progress on the scale of months. And in the 10 months that it took us to go from meeting her to having something that we wanted to manufacture, she was losing clinical milestones. But together with partners like TriLink Biotechnologies and Brammer Bio and Charles River Labs, we did manage to get through all of this in a very, very compressed time frame and receive FDA permission to proceed about 1 year and 6 days from the day that we first encountered her on Facebook. And our publication does illustrate that we saw very promising signs of drug response in association with treatment with our drug, which we've called after her, called Milasen. And this graph shows the seizure response in our patient. Before we started our treatment, she was having between 15 and 30 seizures per day. They were nearly all lasting greater than 1 minute. Essentially, all of them were lasting greater than 1 minute, as depicted here with the dark colored circles. However, as our dose escalations here in the red stars went up, we saw that her seizures came down both in frequency, down to between 0 and 10, 0 to 15, and also in intensity, where we have a transition from dark circles to open circles, meaning that almost none of them were lasting greater than a minute, and typically they were only lasting just a very few seconds. Most of them were so fleeting that the family described you could not tell that they were happening. So this was a response that really surprised all of us. We were hoping for, of course, a response, but I think that this exceeded our expectations. I do want to say that this is a very, very tough disease. I do also want to say that this is not a miracle drug by any means. And in some important domains, our patient does continue to show signs of progression. For instance, her brain volumes did not stop shrinking in association with our treatment, although we believe that they've slowed. However, I think that there's sufficient data here that we've actually been really quite pleased with signs that we're on the right track, that this type of approach is promising and that she is receiving clinical benefit from this, even though it's far from perfect. Now, I want to pause here and comment a little bit about this path, because we are a basic science lab, and even though I am trained as a clinical neurologist, I spend 100% of my time doing research, and most of the work in our lab prior to this point had been basic in nature. So I wanted just to pause and say that in deciding to embark on this clinical project, this is not a clean project. There are many things that were uncontrolled in this project. Obviously, there is no true control in this project, other than the very characteristic and very predictable natural history of this disease. But there are also many other things about this experiment that made it a non-ideal experiment. As a clinical trialist, a seasoned clinical trialist, which I will freely admit I was not, would look for more carefully designed indicators of clinical trial readiness before starting something like this. The lack of there's no biomarker for this particular disease that's been established. There's no way to detect, there's no antibody against the MFSD8 protein, which is missing in her. And so the available measures to us are really primarily clinical in nature. Now, arguably, I'd say that MRI volumes and seizures are very meaningful measures and reasonably objective clinical measures, but I just want to point out that there's also messiness in this too. So how did we decide to actually proceed with it? And I wanted just to point out 4 things here that gave us confidence to treat, and they're relevant because the rest of my talk, I want to talk about extensions of this experience. First, the decision to treat here was really motivated by primarily by the fact that this was such a progressive and devastating disease. What that meant was that of the potential harms that we could easily imagine and write out on a consent form, that we knew that what we were up against were harms that were significantly as bad or worse as harms that we can imagine incurring through our treatment. So the risks of treatment in here normally have to be considered very carefully, and they still have to be considered very carefully, but they're balanced in this case by the risk of disease, which we knew would be progressive neuronal functional loss and loss of quality of life. That might be obvious, but I think it deserves to be pointed out. Second, this is a monogenic recessive condition. It's relatively, as genetics go, simple biology, and the recessive conditions have the added advantage that we know that parents are usually the carriers of the mutations that their children have when they have recessive mutations. And those parents have one good copy, one bad copy, which means that 50% gene function is completely compatible is compatible with normal neurologic function. And so a little bit of gene restoration goes a long way. That, combined with the fact this was a mutation amenable to splice rescue, we knew precisely what the mutation was, we knew the mechanism. It allowed us to really argue to ourselves that the precision with which we could target our treatment gave us a higher confidence, a prior probability that was higher that we would have positive impact. This was less of a shot in the dark, less of, for instance, a small, perhaps different, arguably different than some small molecule inhibitors that you're hoping may have more pleiotropic effects or more indirect effects on a disease state. We were targeting the root cause here. Third, we were working with a well-characterized drug platform. ASOs obviously have been around for decades, and significant investments have been made by others in their biodistribution, their PK, their toxicity profile. And we had at least one FDA-approved template in Spinraza, which at the time that we started this had been given to a few thousand individuals. So that gave us significant confidence as well, because we were not dealing with a complete unknown. And then the last point, we were working with a family with a child with a very rare disease, with even an N of 1 drug. And what that meant was that whatever risks we discussed with the family and were accepted by the family would be risks that they would bear on their daughter's behalf. But this was not a drug to market. And so this really did not have as much of a public health impact as, for instance, any other type of drug. This was really more of an individually consentable intervention, almost a surgery in that respect, because of the lack of immediate applicability to any other patients. As I mentioned, this work was published in the New England Journal in October last year, and it got a lot of attention because a lot of folks are interested in this idea of programmable medicines. This is a concept which has been around since the ideas of monoclonal antibodies, the idea that you have a platform, a technology, whereby different drugs in the same class share many of the same pharmacologic properties, but they differ only for antibodies, the antigen specificity. For ASOs, they differ only in their sequence. And in particular, we pointedly made Milasen, we made sure to use the exact same chemistry as had been deployed for Spinraza, the same backbone chemistry, the same nucleotide modifications, and so forth. So they really truly only differed in sequence and in length. I think Milasen was 4 nucleotides longer than nusinersen. And so our experience has led to a lot and rekindled a lot of these discussions of platform medicines, programmable medicines, and so forth. And, and headlines like this, if DNA is like software, can we just fix the code? I think that it's clearly not going to be as simple as that, but that is the prospect that we are reinvigorated to grapple with. There have certainly been many inquiries since we published our work and since this got coverage from hundreds of rare disease families, clinicians, foundations. And we note that of the inquiries that we get, it's important to stress that we were able to take advantage of a particular type of mutation that created a new splice site. And most mutations don't do that. But the converse, of course, is that some mutations do. And we estimate that about 10% of mutations, up to 10% of mutations in recessive diseases, or up to 10% of patients with recessive diseases may have mutations like this. And bearing that out from the hundreds of inquiries that we've received, we've found a few dozen patients who, like Mila, have orphan diseases, often fatal diseases, and they appear, at least on paper, to be ASO amenable. So the question we've been grappling with in the last 2 years since we since word of this project first started coming out is what to tell these patients, what should we do with these patients? And so we've been incubating this idea that this is something that deserves more attention. That this case, on one hand, in many ways, it feels like a one-off. There are many things that align for us to be able to pursue this case. On the other hand, it does stimulate us to think about using ASOs as a platform for small N medicines. And what do I mean by that? I mean that for orphan diseases that are too small for commercial interest, they aren't attracting companies to come work on this condition, because if you've got only 200 individuals, that's not enough of a market for a drug company to see a path to reimbursement. What are the values of pursuing these? Well, one, they fulfill unmet needs, but two, there are scientific values as well. These types of investigations can be used to validate gene targets and also to probe critical therapeutic windows. No one had ever attempted a therapy for CLN7 Batten disease before, and no one knew, we continue to have insufficient data to know when are the critical windows, when is it too late to intervene, when do you have to get in for optimal benefit. And these types of approaches, small, nimble, rapidly developable ASOs, if they can be used safely, they give us an opportunity to probe these questions. To do this, we have to do it scientifically. We have to do it consistently. We have to keep constant, for instance, I would argue, the chemistry, the mechanism of action, the administration route, because the last thing you want is a series of one-offs. It's hard enough to learn from N of 1 conditions as it is, that you can't have a series of anecdotes for which there are too many variables. You have to use different sequences to change the biological targeting, but everything else as much as possible should be made the same. And importantly, if these are to be undertaken, data has to be shared to aggregate these experiences so that they're more than the sum of their individual parts. We think that intrathecal splice switching ASOs, as we design Milasen to be, are a good place to start. We say that because ASOs can be dosed intravenously, they can be dosed intrathecally. One of the big advantages of dosing intrathecally is that the half-life in CSF and brain tissues is extremely long, on the order of 5 months. And so you can give very low doses if you administer it intrathecally, compared to when you dose things intravenously, where the half-life is more on the scale of 1 month. So the doses in IV are 100 times higher than those given intrathecally, and they're also given more frequently. So this changes your risk profile significantly. Also, splice switching ASOs, which was the mechanism that we used, are non-catalytic in nature. And they are arguably of much more modest off-target risk than knockdown approaches, where there is catalytic activity to bind to a target, to activate RNase H, and to cleave that target. Splice switching works by steric binding and hindrance, and so should be safer, at least on first principles, and empirically, this seems to be supported so far. There are a host of ethical and regulatory considerations that a pilot like this engages, and our article was accompanied by an editorial by Janet Woodcock and Peter Marks, the heads of the FDA, touching on these. What are the standards by which individual patients should be exposed to risks or potential benefits from interventions like this? Where would the manufacturing come from? Who's going to pay for it? A whole host of issues that deserve exploration. But we think that it deserves continued exploration, and we think that those things can be managed. We just have to work through them. I'm sharing with you a case here of a follow-up case to Milasen that we're working on right now. This is a 2-year-old girl with ataxia telangiectasia. And she was actually diagnosed at birth via an abnormal newborn screen. And exome sequencing landed a diagnosis of AT. This is a cerebellar neurodegenerative disorder that is of slower onset than Batten disease, but also debilitating and eventually fatal. These kids are wheelchair bound by age 10 and lose the ability to read or speak because of inability to coordinate their eyes, to coordinate their brainstem movements, and can't swallow either. And we found that she had recessive mutations in the ATM gene, the cause of AT, and one of those was ASO amenable. So this is a second case now that we began working with in May of 2018, supported by the AT Children's Project, a foundation supporting this disease. And we went through screening cell lines, identifying, designing and identifying ASO hits, validating their functionality, selecting leads, manufacturing toxicology, and we began our first dose of this patient in January of this year. And since starting this, well, she's doing well, and we'll comment on her results later as they're available. But we've also identified 2 other patients who have the same mutation for whom this drug could potentially be applicable, and we are working to recruit at least one of them to join this to make this an N of 2 trial as well. So I'm going to wrap up here. I'd say the tools for drug development are really getting quite good. And we think that approaches like this applied to a couple of selected diseases would be an appropriate exploration for pushing this idea further. We've done surveys of AT in particular and looked through a registry of 235 AT cases. And we found 16 cases of those that look like they are ASO amenable. We validated those. We found 10 others that are predicted to be ASO amenable, and there are a few others that we're looking at further too. And we think that going through these, overall the numbers are going to be consistent with between 10 and 15% of this particular disease having mutations that can be addressed with this approach. And so we're beginning to think about actually addressing them. Where all of this will go is unclear. It's hard to say how this is going to scale. There are a couple of different models. Cancer vaccines and CAR T therapies are individualized therapies made in a common way, and the FDA has seen fit to approve those. Is that an appropriate regulatory route for this type of thing? We'll see. Could we keep patients like this under an IND for eternity, never filing for marketing approval and always do them on an investigational basis? We'll see. Or could this go the route of a surgical model, such with an analogy like organ transplants, medically innovative care? We'll see. But we're committed to working in this space with the FDA, with stakeholders like the Oligotherapeutic Society, and foundations like the NLORM Foundation, started by Ionis, to work through this and work this out. So I'll stop right there. I'm a little bit over, but I wanted to end on these 2 acknowledgement slides. First, for the Mila project, and second, for the AT project. Thank you for your attention.
Great, thank you, Tim. We have time just for one question, so I'm just going to ask it. This comes from Marlene Belfort. She asks, has the ASO therapy resulted in any improvements to Mila's vision or neurological symptoms aside from just the seizures? And then the other question, which comes from Niels Walter, is what is the total cost for developing these kinds of drugs?
Right. So with respect to the first question, did it help her vision or other neurologic symptoms? Her vision, unfortunately, by the time we started, at the time when we met her, she had light perception. By the time we started the trial, she had lost light perception too. And so the retinal light detecting cells had actually all died by the time that we started. That's not quite true. She was able to detect ambient light. Her pupils would dilate or constrict in response to room ambient light, but she had no contrast detection left because the only cells that were left were a different class of retinal cells that transduced that. Other neurologic symptoms, in addition, her family reported, but we couldn't quantitate, a striking improvement in her swallowing. And they also reported improvements in her alertness in association with the drug, but we don't talk about that because we couldn't really quantitate. It's harder to say. These are subjective reports. Other things got worse. Her movement disorder symptoms, well, she didn't have movement disorder symptoms to start, but she has developed them. And those are due to deeper nuclei in the brain, and we wonder whether or not the drug might be getting into those or not. Second question, the cost of this, I think I always preface by saying it's premature to talk about cost when you've only done something once and you've done it on a research basis. And we've had lots of volunteered expertise from many people. So in the very early days, as a research project, ballpark, this was in the ballpark of $2 million, actually less than that. But that's a number with very big error bars, and it could go significantly, arguably go significantly down with this type of chemistry and with streamlining the approach.
Great, thank you, Tim. I'm going to end it there. I will send you the other questions, and you can take a look at them later. Or maybe you can just look on the chat and you can answer those questions yourself. So now we're going to introduce, if you could just stop your sharing, we're going to introduce the second speaker of today. So it's a pleasure to introduce Dr. Shobha Vasudevan. She is an associate professor at Mass General Hospital. She performed her Bachelor's of Science in Microbiology and Chemistry at Bangalore University, and then her PhD at Rutgers University, and was a postdoctoral associate at Yale University before starting as an assistant professor at Harvard Medical School and Massachusetts General Hospital, where she's an assistant geneticist. So she's going to be presenting the second talk today, a specialized post-transcriptional program in chemoresistant quiescent cancer cells. Welcome, Shobha. Shobha, we may not be able to hear you. You may be on mute. Shobha, can you go back? I think you're on mute.
Can you hear me now?
Oh, yes. Yeah, that's great. Thank you.
Well, good day again, everyone, and thank you for having us here. In my lab, we are very interested in understanding the mechanisms of gene expression in cancer cells that are quiescent. Quiescence, or G0, is very briefly defined as a transient or reversible arrest from the cell cycle. Such cells are important because they're present as a very small population in tumors, but they are quite clinically relevant, in the sense that these cells are very different in their properties compared to their proliferating neighbors. If you treat these cells with unfavorable conditions, proliferating cells can be sensitive to these unfavorable conditions, while these quiescent cells can be quite resistant. This includes treatment with chemotherapy, and this is what makes these cells very important, because these are the cells that come back and give you a recurrence. So in the lab, we are interested in understanding what are the properties of these cells, what is the gene expression that underlies these properties, and can we target them. So as a model system, we like to use acute monocytic leukemic cells, like THP1, and a number of other cell lines that you might be familiar with, because these are quite amenable to induction to G0 by methods such as serum-starvation. So you can take a mixed cell cycle population of THP1 cells and serum-starve them, and these cells go into a reversible cell cycle arrest, showing markers that are significant for G0. And most importantly, you can reverse the cell cycle arrest or G0 arrest by returning them back to serum when they go back into proliferation. Now, if you take these two populations of cells, the serum-grown proliferating cells and the serum-starved G0 cells, obtained from the same batch of cells, and treat them with anti-leukemic chemotherapy such as citraben or Ara-C, we find that the proliferating cells tend to die in a dose-dependent manner, as you'd expect AML cells do. But the G0 cells, not so much. They tend to be more sluggish in their response, and they persist for extended periods of time. This suggests that these G0 cells have some sort of an apparent chemoresistance. Now we can do this experiment properly. We can take cell lines or even patient samples and treat them with chemotherapy, as you would with patients, and the cells that are left behind are the true chemo survivors. They show a similar IC50 compared to those G0 cells, and at the time of isolation, they show the same G0 markers that we saw with these cells. Now, when you see that two different cell populations that have been derived or treated differently are showing a common property of chemoresistance, you want to identify what is the common gene expression underlying these cells, because that could tell you something about what is important for resistance. So we profile them at every level possible, looking at their RNA levels, looking at their translatome, and looking at their protein by differential label and mass spectrometry. And the idea was to see what was explicitly expressed in these G0 cells compared to proliferating cells, and what was exclusively expressed in these Ara-C chemoresistant survivors compared to proliferating cells. And then see what matched up over here, because that would be similar to the common property of chemoresistance. So we did this at multiple levels, and if you look at it at the RNA level, you see some genes that are in common, but not very much. But when you look at the translatome and the proteome data sets, a significant gene set pops up as commonly expressed in both the apparently chemoresistant serum-starved cells and the true chemo survivors. So what these data told us was that there's specific gene expression in common when these cells become chemoresistant, regardless of how we got them there. And the fact that we see them best at the protein and translatome level suggests that post-transcriptional mechanisms may be involved. So the lab likes to ask two questions. What are the genes that are post-transcriptionally upregulated in G0 resistant cells? This is a bit of a loaded question. We want to know what are the genes. We want to know what they do for chemoresistance. And ultimately, we want to know whether we can target them. And the second question is, how do these cells know to turn up these genes while their proliferating neighbors are producing other genes? How do they know to switch that? And so I'll tell you a little bit about this, but I can also tell you that we're scratching surface. We have only found very, very rudimentary mechanisms, and there's a lot more to explore in the system. So in a nutshell, what I'm going to tell you is that chemotherapy itself induces signaling that is also seen in G0 cells, and this alters post-transcriptional mechanisms. The underlying theme of what we are seeing, at least in the mechanisms that we've looked at, and we have many more to look at, is that post-transcriptional mechanisms that are conventional or seen in proliferating cells are reduced or altered. And this allows non-canonical mechanisms to operate, and when they operate, they turn on specific genes that seem to be directed for tumor survival and chemotherapy resistance. So I'm going to tell you about a story about specific mRNA stability of a class of RNAs due to an RNA binding protein that is affected by this signaling. Then I'm going to tell you that translation mechanisms are also altered, and we see both rate-limiting steps of translation altered over here. So normally you have two rate-limiting steps in translation initiation. The first, one of them is recruitment of the RNA to the ribosome, and this is brought about by complexes that bind the cap and interact with the 3' end. And this is very much altered over here so that you're turning down what happens in proliferating cells and you're turning up what is required for these cells. The other rate-limiting step of translation initiation is recruitment of the first tRNA, and this is also altered over here. And I'll give you a brief overview of that, and hopefully my postdoc will continue talking about that at the Cold Spring Harbor meeting. So to begin with, what we did was we looked at serum-starved G0 apparently chemoresistant cells and the true chemo survivors that we obtained from patient samples and from cell lines, and compared them also with normal non-cancerous cells to find out what was commonly expressed over here but not in normal cells. And when you do that, you see that there are as many upregulated genes as there are downregulated genes over here. It's not all shut down of gene expression. If you look at a snapshot view of this, you see that the RNAs that are polysome associated, which are shown in the middle block over here, are not necessarily concordant with the RNA level changes that we see over here. So we categorize the genes into 4 different categories. Those RNAs that we found were stabilized at the RNA level and then further increased at the translation level, which is the largest category of genes that we see over here. Those that are purely regulated at the translation level or actively translated, where there are no changes in the RNA level, and then those that are poorly translated, and there's a class of genes that are degraded as well. So looking at the largest class of genes, which is inflammatory response genes, this is a very large group of genes, and the top category over here are pro-inflammatory cytokines, and the top gene over here is tumor necrosis factor alpha or TNF-alpha. So TNF-alpha is part of your innate immune response. It requires no introduction. It can also promote NF-kappa B signaling, and that can either promote cell death or it can promote cell survival. And so Charlie, the postdoc who did all this work, was able to show that NF-kappa B signaling is turned on, and it's turning on an anti-apoptotic gene program with the BCL family of genes. So the rationale that we came up with is TNF is a master regulator that is turning on anti-apoptosis genes, and this allows the cells to survive chemotherapy, which is trying to kill it. So a simple method to prove that was to go and get anti-inflammatory inhibitors, and we got these from the rheumatology department at our hospital because TNF-alpha is a common target for autoimmune diseases in the rheumatology world. So we settled on this drug called pirfenidone, which is an anti-inflammatory. People take it for fibrosis. It can target a few signaling pathways, one of which comes up in our in my slides today, but it also targets TNF-alpha translation because it affects the 3' UTR complex. And this has been published much earlier, but the mechanism of how the drug actually affects the 3' UTR complex is not known. Nonetheless, it is known that it affects the translation and the levels of TNF-alpha fall in our system. So if we take our chemo-resistant cells that have been treated with RSC and they are quite resistant, we now treat them with pirfenidone and RSC. We find that almost all the AML cell lines that we've looked at show reduced chemo survival. This is not a cell line artifact because we can test it out in patient samples, and we see again the same phenomena. The treating with the anti-inflammatory also causes a reduction in chemo survival. And we can do this with a couple of other anti-inflammatories as well to prove that this is not just pirfenidone. Normal cells do not produce TNF-alpha unless you have an autoimmune problem, and so they don't respond to it. And finally, to prove that this is actually partly due to TNF-alpha, we knock down TNF-alpha, and we can see that chemoresistance goes down, apoptosis goes up, and you can add back TNF-alpha, and of course, you can rescue the chemoresistance. So this is not to reiterate the point that TNF-alpha is involved in tumor biology. It has many roles in tumor biology, but to show that with chemotherapy treatment, we are changing post-transcriptional mechanisms to turn on genes that are vital for survival for these cells. So we wanted to know how these genes are upregulated, and in particular stabilized and upregulated, which was about 30% of our upregulated genome that we see over here. So we found that if we look at all the 3' UTRs of these genes in both the leukemic backgrounds as well as 3 other solid tumors that we looked at, all of them show enrichment for AU-rich elements. So AREs or AU-rich elements are 3' UTR post-transcriptional regulatory sequences that you may well be aware of. They are very powerful regulators that cause tight and rapid regulation. So essentially in an immune cell, for example, or in any other cell, you find these genes in the 3' UTRs of genes like cytokines, growth factors, and oncogenes. And so these genes are very tightly regulated by the ARE. So in an immune cell that is stimulated by LPS, within 20 minutes, you would have TNF-alpha and other innate immune response genes going up about 50-fold, but they have to be brought back down within 3 hours, and the ARE is instrumental in doing that. Without the ARE, you would have an autoimmune response. And what the ARE does is it binds proteins that cause mRNA decay and translation repression. But the ARE is also responsive to signaling, and so it changes the ARE binding proteins, and you can get stability, you can get efficient translation, and you can increase the cytokine levels. So knowing that, we looked at what could be the ARE binding proteins that bind you, assuming that you would see some of those categories that we see in this green box over here. The first thing that we noticed is that some of the decay complex factors are hugely altered in these G0 cells. So predominantly, we find that the exosome RNA complex is reduced, and this is true not only in our chemoresistant cells or in the serum-starved G0 cells, but in a number of cell lines, and also our colleagues in yeast report the same in nitrogen starvation. And this suggests that this may cause profound changes for 3' to 5' decay, as well as for RNA processing. And this is why we think that we're only scratching surface when we're looking at the G0 program. When we look specifically at ARE binding proteins that cause translation repression or decay, most of these factors, such as the HSRP protein or TIR proteins, are downregulated, which makes sense because the ARE proteins are, the ARE mRNAs are stabilized over here. So we did a broad, unbiased screen of RNA binding proteins using Tim Hughes' dataset to see which proteins are upregulated in conjunction with these ARE mRNAs. And this led us to a really devastating find. We found the least protein that we wanted to find over here, which was TTP or tris-tetraproline. So TTP or zinc finger protein 36 is an unfortunate find because it is the best factor that I can think of for causing ARE RNA decay. So we were quite confounded by this finding, except the TTP field is extremely advanced. And they know in this field that TTP has a phosphorylated form, and when it's phosphorylated, the decay activity is blocked. TTP, when it's phosphorylated, also increases in protein levels, and this is thought to be because of protein stability, as well as the fact that the TTP mRNA itself has an AU-rich element. And because its decay activity is blocked, ARE mRNAs are now stabilized and expressed, and so we see TNF-alpha also going up at this time. Just like in the LPS immune system, we see that the same enzyme MAPCAP2, which is activated by LPS, is also upregulated over here in these RSC-treated cells, and that accounts for the increase in TTP, that accounts for the activity of TTP in this form, and the increase of TNF-alpha. So this was good. We had gotten from our gene expression datasets to cell survival and chemosurvival to phospho-TTP and MAPCAP2. But we hadn't started off with LPS. We had actually started off with RSC, chemotherapy, or serum-starvation. So Charlie was able to painstakingly work out that RSC, which is cytosine arabinoside, a drug that causes replication inhibition and turns on DNA damage signaling, was able to turn on P38 MAPK, which triggers MAPCAP2. and serum starvation turns on p38-MAPK as a well-known G0 marker. So it suggested that chemotherapy itself was turning on post-transcriptional changes that was leading to this TNF-alpha increase and chemosurvival. So Charlie decided to test that using a well-known TDP mutant, which is mutated so that you cannot phosphorylate it. So this mutant is dominant for causing ARE mRNA decay. And when Charlie expressed this, we found that all those genes that were stabilized and translated now had very short half-lives. And if you look at TNF-alpha, the levels fall down because its half-life is reduced. And concurrent with that, we see that chemoresistance is now lost. At the same time as we were doing these studies, there was a parallel study on chronic myelogenous leukemia, which is a different sort of leukemia. And they were looking at drug resistance in patient samples throughout. And they found that as markers, they could use the increased levels of TDP and ARE-bearing mRNAs, suggesting that the mechanism that we see here is not true only for this cancer, but also other cancers. So this was good, and we had learned two things from this. We had learned that chemotherapy itself, which we use, and which often shows resistance in the clinic, was turning on post-transcriptional changes that were actually important for this chemosurvival or chemoresistance that we see here. But from the RNA field, or the ARE RNA field, we also knew that this was a transient mechanism. So the TNF-alpha increase that we see at 4 hours about a 20-fold increase. And we know it's transient because we see the signaling that we see going up over here, going back down in 72 hours. So going back to that experiment with pirfenidone that I showed you earlier, where we got this lukewarm response in reducing chemoresistance, we wondered if this was because we were adding pirfenidone to block this chemosurvival pathway at the same time as we were triggering this chemosurvival pathway using RSC. And so some cells were able to escape the block that we were providing with pirfenidone and get down to this stage and escape regulation by pirfenidone. So to test that, Charlie decided to put in pirfenidone first at various time points before treating with RSC. The idea being that you put the roadblock in first and then you treat with RSC, so that RSC kills all the proliferating cells as it's supposed to, and when it tries to trigger this bypass pathway, it gets stopped at this step. And we should get better or efficient killing with the chemotherapy. Conversely, we also treated with RSC ostensibly to kill all the proliferating cells, inadvertently enrich for these chemosurviving cells, and then try to clean it up with pirfenidone, and we expect that would not work because pirfenidone targets upstream. So this is one of the simplest experiments we've done, but it is reproducible in vitro and in vivo, and it's a beautiful experiment actually, because when we take cells that are chemoresistant to RSC treatment, and just 2 hours before we treat with RSC, we treat with pirfenidone, we find that we can reduce chemosurvival quite drastically down to less than 15%. At the same time, if you do it with a co-treatment approach that I've shown you earlier, we get much less response. And of course, if you come in after RSC treatment, we find that the cells all chemosurvive, but we don't see a significant effect. And if you profile these cells, you find that the anti-apoptotic genes are turned on, and so targeting upstream does not help. So the timing of the post-transcriptional mechanism also seems to be important. So at this point of time, we decided that it was important to target p38-MAPK and turn off TTP phosphorylation, because we knew that it was not just TNF that we were targeting, but also a whole host of genes that phospho-TTP was inadvertently stabilizing. And so we got this drug from the company Lilly, which turns out to be a good p38-MAPK inhibitor, and we could show that TTP phosphorylation is reduced, and alongside that TNF is also reduced. And we could combine that with the pirfenidone inhibitor and get better cell killing. So in every AML cell line that we know exists, we find that if we use these drugs, even at half doses, we find that pre-treatment before RSC treatment can kill all these cells and turn up apoptosis in these cells. This is also true in patient samples that we tested at Mass General Hospital, but not in normal patients, if you will, because these cells do not express TNF and do not express this pathway. Now, this is not all entirely due to TNF. When we tried to examine why this apoptosis was being turned on by the p38-MAPK inhibitors, we realized that when you block p38-MAPK, you turn on the JNK pathway. This is a well-known phenomena, and JNK pathway can turn on apoptosis. And we see that indeed when we treat with pirfenidone and LY, we get the JNK pathway, and we can rescue chemoresistance, if you will, by treating with a JNK inhibitor. And the reason for this is that the TTP phosphorylation not only stabilizes TNF-alpha, it also stabilizes other signaling molecules like DASP1, which is dual specificity phosphatase. This is an inhibitor of the JNK-mediated apoptosis. And because of that, you're blocking apoptosis as well. So in summary, what we see is that RSE, through its phosphorylation of TTP, through this change of post-transcriptional regulation, is turning on different sets of genes. One was the pro-inflammatory cytokines, which promote anti-apoptotic genes, and the other was signaling molecules, which block apoptosis. And so when you come in the clinical world with drugs that block these anti-apoptotic genes, they often show resistance because you're not blocking this arm of apoptosis. And so blocking upstream turns out to be actually a good idea. So at this point of time, we realized that we were only looking at RNA stability, and there was also a translation component to it, and that these RNAs would have to be somehow translationally elevated. We also knew that there were a bunch of genes that were actively translated. That means the RNA levels do not change. And these genes are also very important for chemo survival in vivo. And there are genes like cell cycle genes that are not translated at all and actually suppressed. So to understand this, I'll give you a brief overview about what we think is happening in these G0 cells in terms of translation. So there are two rate-limiting steps of translation initiation. The first is recruitment of mRNAs to the ribosome, and this is brought about by the cap-binding protein EF4E, which is an oncogene that drives proliferation. So EF4E binds the cap, it binds EF4G as well, and this complex together binds the cap as a clump. It brings in EF3, which brings in the ribosome 40S subunit. And 4G can also make connections with the 3' end, which is supposed to enhance translation. Now, all of this is possible when you have the inhibitor of this 4E4G interaction, 4EBP, kept silent by phosphorylation. And this is done in proliferating cells by high mTOR activity, which is a kinase that is associated with growth of the cells. Now, G0 cells have a hallmark factor that there is low mTOR activity. And chemoresistant cells also reduce mTOR activity through much the same pathway that I've shown you earlier. So the DNA damage signaling turns on AMPK, and downstream of that, you turn on these inhibitors of mTOR activity called tuberous sclerosis proteins. And so you reduce the kinase activity that would keep this suppressed. At the same time, the P38-MAPK pathway turns on STAT1, and that seems to promote 4EBP levels. So you're making a lot of 4EBP, but you're not phosphorylating it. So you make a lot of active 4EBP that will disrupt the 4E4G interaction. But I've shown you quite a bit of data that says that there is translation happening over here. It's not that it's translationally suppressed. So there must be non-canonical mechanisms that operate over here. And indeed, if you look at these RNAs that are translated like TNF-alpha, we find that their 3' ends also are quite different from what you'd expect in a proliferating cell. You find that they have short poly-A tails. If you ask how short and measure it, the report is you find that it is shorter than a PAB binding site or a poly-A binding protein site. If you make the RNAs longer, with longer poly-A tails, they seem to need PAB to be removed, and we can do that with proteins like poly-A interacting protein 2, which rips it off the poly-A tail and rips it off from its interaction with 4G. So these cells are really trying to avoid PAB because they want to avoid canonical translation, and they perhaps don't reduce their their RNAs even though they are poly-A shortened because the exosome complex is also suppressed to some extent. So to cut a long story short, we see that chemotherapy signals to the TTP phosphorylation pathway to promote mRNA stability of a bunch of genes that are important for chemo survival. But more than that, it also turns off canonical translation through activity of 4EBP. When you have this active, you cannot have 4E-dependent translation, which Ruggiero and others have shown turns on proliferation. And this makes sense for a G0 chemoresistant cell because you don't want to be proliferating when chemotherapy is targeting proliferation. But it goes a little bit beyond that because 4EBP being active turns on other non-canonical pathways that we are still uncovering today. So earlier what we had discovered is that 4EBP, when it binds 4E and breaks up this interaction with 4G, allows other cap binding proteins like poly-A ribonuclease to now bind the cap. So 4E and 4G together bind the cap in the nanomolar range, and it's very hard to compete with that. But poly-A ribonuclease can actually compete with 4Gs of this complex because it becomes in the same KD range. And alongside that, we find that 4EBP also increases other RNA regulators like this FXR1 RNA binding protein that we had found in this complex that we've talked about before. So FXR1 is an RNA binding protein that associates tightly with a ribosome recruiter that looks like EF4G. And this is a protein called P97DAP5 that you would have heard about from many groups like Nahum Sonnenberg, Bob Schneider, Junya Yamanaka, and Adi Kimchi. And essentially, P97DAP5 is a non-canonical ribosome recruiter because it doesn't have the domains to bind for a binding protein or EF4E. So it really fits the bill for operating over here. And at the time when Irfan Bukhari in my lab published this paper, Yamanaka's group also published a paper showing that the FXR1 P97 complex operates in embryonic differentiation, and Bob Schneider's group showed the same in breast cancer stem cells. So it suggests that there are non-canonical mechanisms that remain to be discovered that the chemoresistant cell is harnessing in order to turn up specific mRNA translation and turn down proliferation. So in summary, it's not just the one of the rate-limiting steps of canonical translation that is suppressed. The other rate-limiting step of canonical translation initiation is also affected. So this is recruitment of the first tRNA, which is brought about by the AF2 complex, AF2 alpha, beta, and gamma. This is subject to a lot of regulation by viral infections as well as stress. And we find downstream of RSC through the STAT1 pathway that upregulates PKR, as well as through ER stress, AF2 alpha is phosphorylated. And this has many consequences on a number of RNA regulators, including that FXR1 protein that I told you about. And if you try to override it, if you come in with drugs to block EF2 alpha phosphorylation or to function downstream of that EF2 alpha phosphorylation to turn back on canonical translation, what you find is that cell cycle genes that are normally suppressed are now turned back up. And so these cells become very sensitive to chemotherapy, and you can actually get better chemo killing, or killing with chemotherapy. So at this point of time, we wanted to see if we could take our drugs all the way to the clinic, and so for that you have to see if it functions in vivo. And we knew that the Lilly drug that blocked P38/MAPK had been tested for toxicity, and the pirfenidone drug is already in people. It's used for fibrosis, and so we used this combination, and we tested out a number of AML mouse models, again with the same method of pre-treating it with pirfenidone and LY first, and then treating it with RSC. And in every mouse model so far, we've seen that it is really efficient at destroying the tumor and at increasing the survival of these mice. So essentially, what these data taught us is that chemotherapy and drug resistance that we see is not just due to mutations in the DNA, but also because of post-transcriptional mechanistic changes. And the timing of these mechanistic changes is important for targeting them to get better efficacy with these chemotherapy drugs. Finally, the most important slide that I have to show you is that of my lab. So much of the work that I told you about today was done by Sunchul Lee or Charlie. He went all the way from G0 back to all the way to those mouse models, and right now we are in talks with the clinicians for starting a clinical trial. And Charlie is an absolute visionary. He's on the job market. He's particularly looking at RNA centers, and just mentioning that as a casual point. But he's actually wonderful, and he's quite the visionary. The other stuff that I told you about is from Irfan Bukhari, who's very good at finding non-canonical mechanisms. He found the mechanism with P97DAP5, and he has a very interesting story that he'll be presenting at the translation control meeting with that EF2 alpha phosphorylation that I told you about. And much of the work in my lab is supported by my long-standing technician and lab manager, Spencer Truesdale, who is intimately involved in all of the projects that I've shown you so far. And of course, nothing is possible without the collaborative atmosphere generated by the Harvard Initiative for RNA Medicine. And in particular, I would like to thank the people who helped me with the in vivo studies, the Heba Maheshwaran lab, without which we could not have done mouse studies because we're not a mouse lab, and people who loaned us their patient samples and mouse models and a number of other facilities that we needed for this sort of profiling. And finally, a big shout out to the RNA community, particularly the people who helped us with TTP, and also with 4EBP and polyribonuclease, without which we could not have understood the mechanism that I told you about. And thank you for hosting us. I'd be happy to take questions.
Great, thank you, Shobha. Are there any questions for Shobha at this point? Feel free to unmute yourself if you'd like to ask a question, or you could type it into the chat. I know we're a little over time, but we'll keep the meeting going so people can ask questions if they wish. Maybe I'll start and just say, first of all, very interesting story in that, you know, a completely new paradigm for sort of overcoming drug resistance. And you really just sort of touched on, I mean, you touched on so many molecules there, but there must be other potential targets that you can think of in those pathways. I know that, for example, the Sonnenberg lab is already targeting some of those EIF factors, but are there other things that you can think of that could be potentially useful to go after in this paradigm?
I think so. So we looked at RNA stability because we fell into it in the story that I told you is exactly how we emerged with TTP. And we looked at translation as well because we look at translation all the time. But there are many other processes, many other post-transcriptional mechanisms that are altered here. So there's splicing changes, the exosome has changed. So there are many molecules that actually now become very important and were perhaps not important in normal cells or proliferating cells. And so these have great potential to be targeted, but we need to work out how these mechanisms are affecting chemoresistance, because none of this is actually known. There's very little post-transcriptional work in quiescence or chemoresistance per se, at least in this model.
And then just one more question from me. Well, actually, let me ask the other question that's come up. So Ethan Greenblatt asks, do you know if you're inducing the cells to change their translation patterns, or are you selecting for slow cycling quiescent cells when you add the chemotherapeutics?
That's a great question. This is the chicken and egg question that we always get, because when you look at a tumor population and you look at them, they're pretty heterogeneous. You always see slow proliferating G0-like cells within the tumor, and they do have these mechanisms on if you stain them, for example, for F12 phosphorylation or dephosphorylation of 4-EBP. So there might be a low level of cells that are already present that are being enriched and enhanced because you're further turning on this pathway. We can't actually address that from this. We would need some sort of a timing inducible system perhaps to address it. That's actually something that we should address though. It's a great question.
Or maybe single cell sequencing at some point might answer that.
Something like that would be wonderful. Single cell translation control would be wonderful.
Yeah, you just have to invent that. So then another question from Shamoyi Mukherjee. Hi Shobha, wonderful seminar. I'm curious, have you looked at any other ARE binding stabilizing proteins other than TTP, which may be having a role?
Yeah, so initially we looked very hard. We wanted it to be an HUR-like protein. We looked so hard. And then we ended up with, it is TTP and the phosphorylation. And I think at least in this system, it is TTP and phosphorylation of it. And the beauty of this is that it is very malleable, because TTP is such a good degrader of ARE messages. To just have this slight switch with phosphorylation actually gives the cell the ability to go back and forth between being chemoresistant and quiescent, and then maybe months later when you've removed chemotherapy from the patient, it now goes back on to becoming a full-blown proliferating cell. So we did look very hard for as many ARE binding proteins that are stabilizers, and none of them played out in this system at least. But there are other cancers also that we're looking at, and we have the data sets in the paper that is published in Genome Biology. So they may play a role in solid tumors, for example, and that is something to look at. It's a good question too.
All right, well, I'm going to thank our two speakers and thank the audience for attending. I'm going to leave the Zoom meeting open for a little while if people have other questions they just want to ask. Maybe Shobha and Tim can just stay on for 5 more minutes or so, and if there are other questions. Otherwise, thanks everybody, and we'll see you all soon, hopefully. Bye-bye. So if you just want to look at the chat, you'll see people are raving.
Yeah.
So great.